Good afternoon. My name is Ori Rothstein, and welcome to the LiVE1 Knowledge Institute in St. Michael's. So I have a couple of jobs here. The reason I'm here today is because I'm the Associate Director of Basic Science Research at the LiVE1 Knowledge Institute. Most of you who are from outside the hospital probably haven't been here before. It's a relatively new institute. We've only been here for a couple of years now, and I think that the big idea around the Research Institute is it would be a very good way to bring fundamental research through to clinical care in patients. And so that's really the focus of the Institute, but also the focus of a lecture series that we call Popular Science. And here at the Research Institute, we have individuals who do very fundamental research at the cell and molecular level. Oftentimes they take it into animal models to really study the basis of disease. And one of the big hopes that we have is that with that bridge that goes between the Research Institute over into the hospital, that actually we're going to be able to facilitate moving new ideas that we generate in our research laboratories into patient care and really doing first-in-man types of studies to improve care and outcome in the patients that we have. And we've had a broad range of lectures here. They really focus mostly on what our scientists do, and we're really lucky today to have an individual named Sunit. So Sunit's been here at the University of Toronto and St. Michael's Hospital for almost 3 years now. And he's actually a neurosurgeon by trade, so he's a brain surgeon. If you go and talk about it after, you can always say, well, it's easy, it's not brain surgery. So that's what you can say about his. And so Sunit's a really unique guy. From my standpoint, we had the opportunity to recruit him from the United States. So he was at Northwestern University there and came here at St. Michael's Hospital here in our Department of Surgery. He was actually the first person who came from the United States back to Canada to do his career as a neurosurgeon. But he's really unique because he's not only a neurosurgeon, he's also a scientist. And before he actually did his neurosurgery, he actually did his PhD in neurobiology at one of the top laboratories in the United States. So when he came here, the expectation was that he was going to do his research at a pretty fundamental level, and you'll see it today, but also, and he'll also articulate this to you, he's actually going to take that research, hopefully, and take it on to patients. And that's really what we hope he'll do. He's also, we won't tell you that he's a star basketball player. He played for Michigan in the NCAA, the year they actually won the national championship.
Runner-up.
What?
Runner-up.
Runner-up in the national championship. But that's closer than I've ever gotten, I can tell you that much. So without much further ado, I'd like to welcome Sunit and welcome all of you here. We have a pretty broad-ranging group. We have volunteers from the hospital. We have some people who are undergraduate medical students at the University of Toronto. I know that we have 2 high school groups that are here. We have one from St. Michael's Choir School, so that's those guys in the back row there. And then we have a group of guys from Upper Canada College. So what we want you to do is, if you're from high schools, go back to your school, tell everybody how great this is, have everybody want to come, because really the purpose here is, number one, to learn a little bit about science and interesting science and how it in fact influences disease, but also to give you an opportunity to see what a career in science and medicine might look like to you. So without further ado, Sunit Das, and welcome to the podium.
Thank you, Ori. So I did play basketball. It thankfully didn't work out so well, and thus I became a neurosurgeon. If you can't play basketball, you may as well do something else with your time. So like Ori said, I came here from Chicago, and hopefully what I'm going to show you today makes sense of why that's the case. And what I'm going to show you is absolutely a St. Michael's Hospital and Li Ka-shing story, and absolutely dependent upon that. So I've titled my talk From Dark Matter to Target, LiVE1 and Glioblastoma, and hopefully by the end of this, every one of those terms will make some sense to you. So I'm going to start with this. This is This is an old friend of mine named Sid Gertzman and his wife Anna. Sid was a South African who came here to Canada in 1969. I came here to Canada in 2010. I came for a job. Sid came because the South African government decided that he was a terrorist and couldn't be kept there. The reason he was considered a terrorist was because he was fighting against apartheid. When he came here to Canada, he met Anna, who was an oncology nurse, and they had a beautiful daughter, made a life here. I met Sid actually the first month that I came here to St. Michael's Hospital. He was brought to hospital dying, basically, with a massive hemorrhage in the brain. And it turned out that the hemorrhage that occurred was into a brain tumor, something called glioblastoma. Fortunately, he did fantastically well with surgery. He passed away about 5 months ago now, after chemotherapy and radiation. And that's the story for all of my patients with glioblastoma. So glioblastoma is a primary brain tumor. What I mean by that, it's a cancer of the brain. It's not a cancer that arises from the body and travels to the brain, but one that arises from brain cells itself. It accounts for about 15% of all tumors that we find in the brain. And of primary brain tumors, of tumors that arise within the brain itself, it accounts for about 50% of them. It's not common. It only affects about 15,000 people a year, but it accounts for 15,000 cancer deaths a year. And what that tells you is that being diagnosed with this disease is a death sentence. People live about 1 year. That's despite aggressive therapy with surgery, chemotherapy, and radiation. These are often young, healthy people. And if you look at all cancers that we take care of as physicians, glioblastoma has the highest ratio of potential years of life lost relative to any other cancer. So this is what's frustrating about glioblastoma. When I see patients with this disease, what I see in the brain is a large mass. We know by the nature of this tumor that there are cells that have traveled beyond that mass into the brain, but I can remove the whole of this mass off. My patients go through aggressive surgery. They're treated with chemotherapy and radiation month after month on MRI. We don't see anything that looks worrisome. And inevitably, at 7 months, we come in to review an MRI and the disease is back. So these patients, for whatever reason, all of these patients achieve a remission. What we see on MRI is that the disease has disappeared, and all of them succumb to this disease. It recurs in every one of these patients. So this leads to two things for me. One is, it teaches me that as a surgeon, my job is meaningful, but it's limited. And second of all, it tells me that I need to think of this disease as a scientist. So in science, you develop hypotheses that try to make sense of what you're seeing. And the hypothesis here, if you think of these two things, must be that there's some small population of cells in these cancers that are able to resist the therapies that we have and that drive disease recurrence. So we think we found that population. It's something, we call them cancer stem cells. So this is actually something that was found here at University of Toronto by a gentleman named John Dick looking at leukemias. What Dr. Dick found was that if you took all the cells in a leukemia, it turns out that only a small population of the cells are actually the ones that create all the rest, and that when you put those cells from a human into a mouse, it could recapitulate the whole disease. They call these cancer stem cells because they look like normal blood stem cells. They're cancers, but they have the properties of normal blood cells. The interesting thing is that if you look at stem cells anywhere in our body, and all of us as adults have different stem cells throughout the body, those stem cells are often highly resistant to chemotherapy and radiation. They're built to last. And these cancer stem cells have those same properties. So the hypothesis is that glioblastoma recurrence is driven by cancer stem cells. And if you look at this cartoon here, it leads to a second hypothesis, which is that if we're going to actually achieve meaningful remission in this cancer, we're going to have to find therapies that don't only kill 99% of the cells that aren't cancer stem cells, but that also kill the 1% of cells that are able to recapitulate the whole tumor. So we need treatments that are specific for cancer stem cells. So this leads to the University of Toronto and St. Michael's Park. So this is Phil Marsden. Dr. Marsden is professor of medicine here at St. Michael's and University of Toronto. He's the head of nephrology, and he's a big deal. So I'm at the start of my career, and I like the work that I do, but I'm very young. There's a lot of young people here in the audience, so it's not a general thing to go up to someone who's very high up in the world and say, well, this is my idea, right? And so I kind of hold Phil Marsden in awe. And it turns out that Dr. Rothstein was supposed to be part of a lunch symposium where an editor from a big scientific journal was coming, and Dr. Rothstein couldn't go. And he asked me to go instead of him. And I got to this lunch, and the people who were there were the people you would expect at a lunch like this, that Dr. Rothstein and not me should be at. And one of them was Phil Marsden. So being at this lunch, I had to do what any young person who doesn't belong at a table like this has to do to get by, which is start drinking heavily, right? So after a couple of drinks, I got up the courage to ask Dr. Marsden, what are you working on in your lab? And he said, we're working on links. So adding to the confusion of being slightly not on my game, what is a link? So this is the central dogma of biology, and this is from work that was done by Watson and Crick when they discovered DNA. Our understanding of biology is that information in biology goes on in the cellular level from DNA to RNA to proteins. Right? All of us have DNA that are in our chromosomes. That DNA is translated into RNA, and that RNA then serves as a template for proteins that are the workers in our cells. Marshall Nirenberg, a biologist from the 1970s, said basically DNA makes RNA makes protein, and that's the direction things go in. This led to many of us thinking that the most of what's present in the human genome is junk. So if you look at the human genome, only about 20% of the genome actually encodes for RNA that becomes protein. The other 80% doesn't. And we thought, as we always do when we don't understand what we're looking at, that it was all nonsense. So it turns out if you look at that other 80% of the genome that does not make RNA that makes protein, it makes RNA that actually has remarkable importance controlling the rest of the genome. So that 80% of the genome that makes RNA that doesn't become protein, those RNA molecules control all the rest. And we've called them long intergenic non-coding RNA. Non-coding because it doesn't become protein. Intergenic because it's between what we know as normal genes. Just to be clever, they're called links. So this is what Dr. Marsden studied, links. And his fundamental question, his interest in these molecules, was because he's a nephrologist. And what he sees are patients who have diseases of the kidney, often due to dysfunction of blood vessels. And he has a fundamental scientific interest that complements what he does as a physician, which was, what is it that makes an endothelial cell? So think about this for a moment. All of us start off as single cells that then double, then become 4, and then on and on. And somehow the genetic information that's the same in every one of our cells gives rise to the tremendous diversity of all the different organs in our body. The way that happens is that a liver cell compared to a brain cell, even though it contains the same genomic information, the genes that each of those different types of cells turns on and off is different. And that gives rise to different identity despite it starting with the same material. So there must be a particular imprint that cells use to become endothelial cells, or the cells that give rise to blood vessels. So he wanted to look for what that imprint might be. And what he did was something called a gene array. So when I was a graduate student, which wasn't that long ago, we used to get very excited if we could do something called PCR to to measure the levels of a single gene in a cell. And now we have technology that allows us to look at thousands and tens of thousands of genes at the same time. And that's what this map that you're seeing is. This is called a heat map. And what it does is take RNA from a cell and measure the levels of multiple different products and give us a map of what those are. And what you can see here is Dr. Marsden has grown endothelial cell precursors, which were taken from the umbilical cord of babies born here at St. Michael's, and used that material to look at what the gene expression in those cells are. My son was born here at St. Mike's, and I remember signing the consent form for this, so these may be more important to me than I even think. Using that array, he found this molecule, something that he called link RNA VEGFR1, or LiVE1. This was a link, a non-encoded RNA molecule that he found that was near a gene that encodes for a molecule called VEGFR1. VEGFR1 stands for Vascular Endothelial Growth Factor Receptor 1. So VEGF, or vascular endothelial growth factor, is a signal that our cells make in order to promote other cells in the area to become blood vessels. This was the receptor for that molecule. And it turns out that this link was just next to that molecule. And when he studied that link, LiVE1, what he found was that LiVE1 is only expressed in these two types of cells that are endothelial cells. When he looked at other types of cells, it's not there. And the interesting thing was that when he tried to understand its function, what his lab did, this is a spherule of endothelial cells. So again, those umbilical endothelial cells. And if you put them in culture, they'll start doing what they're meant to do, which is make new blood vessels. They start forming these little tubes. And I'm sorry, it's not easy to see here. When his lab manipulated the LiVE1 expression in these cells, so they used the mechanism to knock down LiVE1 or to stop LiVE1 expression, those cells are no longer able to make tubules. So they can't do what they're supposed to do. So this and a few other experiments that they did basically led them to determine that LiVE1 is expressed in these endothelial cells, and that its expression is part of what allows these cells to mature into blood vessels. So this was basically what I recall of our conversation. Phil was telling me about this work, and it's remarkably exciting. And he said to me, we think we found something that's responsible for vascular maturation. And again, you have to remember I'd had a few glasses of wine and felt a little capable of speaking to Dr. Marsden in a way probably I wouldn't otherwise. And I asked, have you looked for this in any vascular cancers? And I thought, he's a kidney doctor. People with renal cell carcinoma or kidney cancer, that's a very vascular cancer. And he said, no, not yet. That's a great idea. I said, don't you work on a vascular cancer? I thought, yeah, I do. So this is glioblastoma. This is what glioblastoma looks under the microscope. And one of the signs that we look for to determine whether or not a tumor is glioblastoma is something called microvascular proliferation. That's a fancy way of saying that one of the necessities of glioblastoma is to make new blood vessels. In fact, that's intrinsic to what glioblastoma is. It has a remarkable capability of making new blood vessels, something that makes surgery technically difficult for us, and something that is absolutely part of the identity of this cancer. And so we looked. And when we looked this is looking at tumors of patients of ours here at St. Mike's, what we found was that LiVE1 is expressed in these cancers, and in fact, thousands of fold higher than it is in normal cells. So just to step away from this for a moment, these are two papers from colleagues of mine in New York and Milan that came out about a year ago that really changed the way we understood these cancers. So glioblastoma needs to form new blood vessels in order to grow. In fact, one of the chemotherapy agents that we use to treat it is called Avastin. Avastin doesn't actually attack cancer cells, it attacks the ability of these cancers to make new blood vessels, and it temporarily works. We had always assumed that the way this cancer makes new blood vessels is by recruiting stem cells from the bloodstream that then can integrate into the cancer and then mature into new blood vessels. These two groups showed that that is one mechanism that's used for blood vessel formation, but that these cancers, glioblastoma, actually have a secondary mechanism. In glioblastoma, the cancer stem cells themselves are able to give rise to blood vessels. So the cancer doesn't even need to use normal mechanisms for making blood vessels. It's able to give rise to new blood vessel cells themselves. So we postulated that maybe LiVE1 is what allows these cancer stem cells in glioblastoma to become endothelial cells. And we looked, again, these are patient samples. This is something called fluorescence activated sorting, where we look for markers on the cell surface that allow us to distinguish between different populations in the cancer. This 133 positive, 144 positive population is a cancer stem cell population, and you can see that LiVE1 is expressed at very high levels in that population. So in other words, LiVE1 is expressed in glioblastoma at very high levels, and it's expressed by the cancer stem cells. And in fact, when we put the glioblastoma cancer stem cells in culture, they start making tubules just like the endothelial cells do. And when we knocked down LiVE1 using the same approach that Dr. Marsden did to knock it down in the endothelial cells, it stops them from doing so. So we were pretty excited by this. Basically, what we'd found so far suggested that this molecule that's involved in the normal processes by which the body gives rise to blood vessels is also present in this cancer at very high levels, and that in the cancer, it looks as well as if it's involved in allowing the cancer cells to act as if they're blood vessels. So we used now an animal model to see whether or not that's the case in the cancer itself. So what we do here is take tumor tissue from the operating room, walk it across the bridge, dissociate it, and implant it into mice. And actually we do it into the brain of our mice. And what we did basically in these models, we call it the xenograft, is in some of these mice, we used a delivery mechanism that allowed us to pump a knockdown molecule against LiVE1 into the tumors. And what we found got us, again, very excited. The red here are tumors that have basically been treated just with a control knockdown molecule. So in other words, they're expressing LiVE1 and they're not being treated. And these are what the cancers normally do. They grow. The blue are the cancers that we treated with the knockdown of LiVE1. So in other words, when we knocked down LiVE1 in these cancers in mouse brain, it caused the cancers to regress. And this is what they look like under the microscope. So this is what these tumors normally look like. And the ones that we treated with the molecule against LiVE1, you can see, look unhealthy. And when we looked at them microscopically, what we found was what we expected to, basically, that LiVE1 knockdown in these tumors resulted in a loss in microvascular proliferation. So in other words, the tumors could no longer form new blood vessels. And this was what was most exciting to me. There are hypotheses that the microvasculature serves as a niche or a home for cancer stem cells. And when we looked for cancer stem cells in these tumors, what we found was that their number decreased tremendously as well. So in other words, knocking down LiVE1 in these cancers kept them from making new blood vessels. It disrupted their ability to form these microvascular niches, and losing that niche caused a loss of the cancer stem cell population. So we're pretty excited about this. In medicine, we always, and particularly in academic medicine, we always talk about going from bench to bedside, of taking what we learn in the laboratory to the clinic. And it's one of these things we all write in our grants. Monday is CIHR grant day. It's something we all put in there. You rarely have an opportunity to feel like there might be some reality to it. And I feel like that's where we are. So what we know that LiVE1 is a cellular effector molecule that's required for endothelial cell maturation and for vascular network formation, for formation of new blood vessels. We know that it's expressed in glioblastoma and that it allows cancer stem cells in glioblastoma to become neoplastic blood vessels. We know that knockdown of LiVE1 disrupts the vascular niche in glioblastoma that's required for cancer stem cell survival. And we think that LiVE1 could be a therapeutic target in our patients. So we've been lucky that we're not the only ones who think that's the case. So you may know the University of Toronto has a collaboration with a group called the Mars Discovery Unit. Mars has taken great interest in this. We're actually completing our patents later this week. They've been securing capital investors for us. It takes about, it's thought it takes about $1 million in 9 years to move something from this stage to clinical trials. But that's what we're getting ready for. So I have to thank these people. I present this as if it's my and Phil's work, and the reality is that this was driven by a remarkably talented graduate student in Phil's lab, Jenny Wang, and a remarkably talented senior manager in mine, Megan Wu. We've gotten great help from Jason Karamchandani, who's one of our collaborators here in neuropathology, and from other colleagues here at Li Ka-shing. And none of this could have been done without our funding help from the NIH and from CIHR and Brainchild. So any questions?
I just had one about when you treated the tumors in mice with the knockdown LiVE1. Did you, I don't know if the lifespan of the mice were knocked out, but were you able to stop treatment or see whether after the tumor regressed to see if it came back?
No, so that will have to be part of the preclinical testing. What we saw was that there's a modest improvement in survival and a more significant improvement in what's called progression-free survival or progression of the tumor. Part of that is because we only treated for a short interval of time. And part of that is because we don't put the mice through surgery before they're treated. And you can imagine what you would use this for is that as a surgeon, I would resect the whole of the solid tumor, and that this would be therapy for the cells that are remaining.
How long ago did you play for MSU?
So I didn't play for MSU. I played for University of Michigan.
Oh, okay.
Yeah. And
I was going to ask you if you
19
shits and Ekpe Udo.
I'm a lot older than that, unfortunately.
Okay.
When you see, when the cancer comes back in patients post-surgery and chemotherapy, do you see development of those vascular niches again?
So if you look microscopically, yes. So and it's interesting, so patients who have recurrence of their disease after they're treated with Avastin, which is a drug that's meant to inhibit new blood vessel formation, it looks pathologically quite different than what disease recurrence looks like otherwise. So that's a great question there. And it looks like there are some cancer stem cells that manage to create a different type of niche that doesn't require that microvascular proliferation. And so that's part of the difficulty. So it's interesting, when I tell my patients about this and they see these results, their question always is, why is it going to take so long? And what I try to explain to patients is that the process that we have in place, the reason it is what it is because one of the overriding purposes of that process is to protect my patients from my optimism. Right? And there may be unforeseen things of what to expect from the biology of glioblastoma treated with this that I'm just not anticipating. Right? It may be that they find a way to recur that no longer depends on making those niches that we know about.
My question is actually kind of a follow-up to that. Are there any concerns for types of side effects that you might have with injecting something like LiVE1?
Yeah, so it's interesting. One of the difficulties of treating brain cancers is that it's hard to get things into the brain because of the blood-brain barrier. One of the benefits is that if you manage to get it into the brain, the risk of systemic side effects because of leakage is also low. But it's one of the things that we'll have to think about during preclinical development. I suspect what the delivery mechanism for something like this will eventually be is actually a virus that's tropic for cells in the brain.
Has that ever been used before?
So there's a group at Duke that is doing preclinical testing with a virus of that sort. It's not a virus that employs inhibition of a molecule, but there's a great interest in developing viruses that could be vehicles for multiple different types of therapy. Yeah.
I'm just wondering, like, based on your response, I know you mentioned that there's a vascular niche that maintains the stem cell population. Is the stem cell population actually responsible for that niche, or does the niche create, maintain the stem cell population?
What are you doing for the next 3 years?
And so, like, I was curious, with the Vascular treatment, does that decrease that vascular niche and then also affect the stem cell population?
That's a great question. It's been hypothesized that there's at least 2 different niches in glioblastoma. One is the vascular niche, another is something called the hypoxic niche. And the stem cell population that we think resides in that second niche is quite different than the one that resides in the vascular niche. Cancers are adaptive. And remember, part of the biology of cancer and what makes it difficult to deal with is all cells are adaptive, but the timeline for that in a cancer is so much faster because their cell cycle is so quick, right? So cancers will evolve to try to exploit the environment that they're in. So when you ask which element is driving, is it the stem cell that's driving that environment, or is it the stem cell that's reacting to that environment? I think sometimes it's hard to tell. But yeah, if anyone here is interested in doing a doctoral project looking at that question, I'm happy to talk after. Great, thank you, everyone.